Wonderful. Okay, let me hand out a little sheet for this one. We're going to stay out of biochemistry for a while. Talk about cell division. Now, when we think about cells, we know that cells are limited in size by diffusion. We found that, for example, because of the relationship between volume and surface area, if we increase the volume of a cell, we don't increase the surface area at the same rate in the same amount. Eventually, over time, what happens is as the cell gets bigger and bigger, then it starts having problems with getting enough exchange with the external environment. And diffusion becomes very, very slow and inefficient. So, rather than cells growing by enlarging and getting bigger, cells grow just by dividing, by increasing the number of cells. And so, in this case, we're going to talk a little bit about this process of cell division, both in prokaryotes and eukaryotes. Now, as you know, cell division is necessary for three main functions. Number one, if you're a single-celled organism, this is the principal ways that you reproduce. You reproduce simply just by mitosis. So mitosis is a very, very common form of reproduction. In fact, a lot of single-celled organisms, this is their main type. Now, one thing we'll find out is when we go and talk about sexual reproduction next after the holiday, we'll find out that even though single-celled organisms reproduce primarily asexually just through cell division, we find that most and virtually all organisms, even if they're single-celled, occasionally do sexual reproduction, where they exchange DNA. And bacteria are just masters of this. So, but for the most part, 90% of the time, these single-celled organisms are simply just making clones of themselves that have exactly the same genetic endowment. For multicellular organisms, things like fungi and algae and plants and animals, we find that instead of actually going and getting bigger, we just add multiple cells. That's what makes us multicellular. So growth is really a product of just adding more cells. The other thing that we find is that cell division is often about repairing things. We know, for example, in our body, we have certain tissues that are constantly being recycled, and new cells are being placed. Lots and lots of renewal. For example, our skin, from the time a new skin cell is made to the time that it sloughs off on our epidermis, is about a month, 3 to 5 weeks. If we think about our digestive tract, if you went and looked at the lining of the digestive tract, you would find that the cells there last about a week, 3 to 6 days. Same with the small intestine and the large intestine. They're constantly being on a little recycling. We get new taste buds every week. We get new smell receptors every month. Even our bones are recycling. A femur will completely replace itself every 10 years. And that's a big, big bone. Right? Our blood is constantly being recycled. Your platelets have a lifespan of about 10 days. White blood cells, believe it or not, some are lifespans measured in hours, and some will last you your whole entire life. Red blood cells, 3 to 4 months, but they're constantly getting recycled. I often think about the body as kind of like that old Buddhist proverb that you can never step in the same river twice, because the river is constantly moving. All right, And just like that, our bodies are constantly recycling and building new cells. So lots and lots of renewal. Of course, one of the great things, imagine having a coat or a shirt or a pair of pants, or for me, the ones that always get holes in it first are going to be my socks. So I always dream about having socks that will self-repair. All right. Our skin can do that. Our body tissues can do that. If you have a surgery or if you have a cut, all right, those parts will come together and seal back up. And that's done by cell division. Now we're going to start off with cell division that takes place within bacteria. This is prokaryotic cell division. And this one has an interesting name. We don't call it mitosis. We call it binary fission. Now that's fairly simple to decipher. Fission just means to split. And of course, binary means two. Think about the binary code in computers. You know, zeros and ones, on and off switches. All right. So it literally means to split into two. Now, how it works is a little simpler than what we find in eukaryotic cells. Generally, and this is a nice little E. coli bacterium, and you can see these are rod-shaped bacteria. And you'll notice that we have some that elongate. And once they get about twice their original size, then they form a wall. This is called a septum. Septum just means a partition or a wall. And then that septum completely goes and separates, and then we get two daughter cells. Bacteria have this remarkable ability to do this incredibly quickly. All right, we'll find out how long it takes in eukaryotes, but generally some of them can do it in less than an hour. It's almost like lens crafters. You know, we'll get you a new pair of glasses in under an hour. Now, since they just have a single circular chromosome, all right, obviously the first thing that has to happen is the replication of the DNA. And if you remember, we have a single origin. The replisome goes in opposite directions until we come to a terminator. And as you can see, we get two copies of the DNA. Then something very interesting happens. We often think about growth taking place at the ends of cells, but in this case, the growth actually takes place between, here in the middle. And as we grow, it goes and separates the two chromosomes. And eventually we get about twice the original length, and then comes the proteins. We have a beautiful little protein that has a strange little abbreviation. It's called the FTSZ protein. And what you're seeing here is our little protein. This is the FTSZ that facilitates the process of forming a partition or a wall, right, in which a new cell membrane and new cell wall material begins to grow and forms a septum. When the septum is complete, then the cell pinches in two and the two daughter cells are formed. Oftentimes in cell division, we call the original cell, we call it the parent cell, and then the two cells that arise from it as the daughter cells. So parent cell and daughter cells. Now, just to kind of show you this beautiful little protein here, I have a picture where they have tagged it with a radioisotope so it would show up on X-rays, on X-ray film. And so here it is. This is a bacterium, and you can see, and it's almost midway through binary fission. And I love this one because it's a little bit of an angle, and you can see the circular nature of it. But this is the FTSZ protein that facilitates the production of a septum. So prokaryotic cell division All right, a little bit different from what we find with the complicated mess that happens in eukaryotes. Now, generally, when we describe mitosis in eukaryotes, we usually describe two kingdoms. We show it how it works in animal kingdom, and we show how it works in plant kingdom. Unfortunately, we have two other groups of organisms. We have, of course, the fungi, and we also have the protozoans and the algae. They have slightly different versions of this process. And your textbook has a nice little comparison of the process of mitosis in those other groups. And some are pretty weird. In fact, one of my favorites is fungal cells. Fungi generally do the entire system without ever altering the nuclear envelope. When we look at plant cells and we look at animal cells, that's one of the first things that happens is the nuclear envelope will disintegrate. Not so in fungi. In fungi, it does it intact. It's pretty amazing. So let me show you this little thing from the textbook. And here's binary fission here. Here's animal mitosis. Right. They're not showing plants. I think may have cut plants out there. But if we look at fungi, all right, the membrane stays intact. All right. Here is some protozoans and some algae. They have some pretty strange ones. In fact, you'll notice that their nuclear envelope stays intact as well. Now, if you're wondering about the name mitosis, it comes from a medical student. A medical student who in 1879 was looking at some mitosis in a plant cell. Not a plant cell, excuse me, in a salamander, in a newt. The cool thing about them, the nifty thing about salamanders is that if you cut off a limb of a salamander, all right, it will completely regenerate that limb. It will regenerate that limb in 7 weeks. Right? And if you keep cutting it, it'll grow back. It'll grow back. Right? So, the cool thing about it is, in places like that where you have active cell division, you can actually go and witness mitosis going on. Now, what he was looking at was the chromosomes. The chromosomes appear, and they look like threads. And that's what mito means. Mito means thread. So, literally, it's the condition of the threads. And we see that because we only see the chromosomes when a cell is dividing or about ready to divide. Oop, I always grabbed the wrong one. All right. Now, to understand how mitosis works, let's talk a little bit about how DNA is packaged. DNA is an incredibly skinny and long molecule. Right? In fact, if we went and connected all the DNA together from one cell, we would have a thread 3 feet long. Now imagine having a thread that's 3 feet long, and you have to put it in a container that's only 5 micrometers in diameter. 5 micrometers in diameter. That is tiny. We know that the best way to do it is just how we organize wire. It's how we organize thread. It's how we organize rope or cable. We wrap it around things. Right? So if you want to compact it and organize it, a great way to do it is to wrap it around things. So that's what we do with DNA. So here's DNA. And you'll notice that we have some beautiful little proteins. These are called histone proteins. Histone proteins. Histone proteins, and you can see there's a variety of different kinds, act like a spool or a spindle, and the DNA wraps around them, right? Just like you would go and use a spool to organize and pack a twine or thread. Now it turns out that in those little coils sort of grouped together, in fact we'll see a little structure called a nucleosome. Right? And if we look at the loosely or diffuse mass of coiled DNA, we just call that chromatin. Right? Kind of looks like slightly organized spaghetti. Now, when a cell is about ready to divide, one of the first things that happens is that after the DNA is replicated, the chromatin will coil and supercoil and produce our chromosomes, our X-shaped chromosomes. This is exactly the same stuff as this, but it's just more coiled. Now, we have some additional proteins that go and help with this process. So let me show you what that looks like here. So we'll begin by looking at, these are the DNA wrapped around our histone proteins, and it forms a little structure. This is called a solenoid. If you look at a solenoid in a car, right, or a solenoid in an electric motor, you'll find what it is just a big coil of wire. And it makes perfect sense that they call these solenoids as well. It's just a big coil of wire. Right? Now, you'll notice there's another protein that's involved in this, and this is what we call a scaffold of protein. Right? A scaffold protein. The scaffold protein goes and starts forming chromatin into loops. And you might have noticed when you were looking at chromatin, you saw these little loops there. And the beautiful thing about it is that these loops go to make a beautiful little thing called a rosette. The nifty thing is we find rosettes also in prokaryotic chromosomes. They form that same similar structure. And you can see our little scaffolding protein. Right? Now, when we get into a chromosome level, these will start coiling and generally coiling some more and eventually becoming our chromosome. Now, what actually makes the DNA stick to the histone protein in the first place? Well, it all has to do with charge. Right? Turns out the DNA has a negative charge to it. So if you were going to make a protein that would stick to DNA and you wanted to make it a positive charge, what type of R groups would you have on the amino acids? Basic ones, right? Yep. So you want to have basic ones. These are things like arginine and so on and so forth. And that's exactly what we do. These are rich in basic amino acids. And it gives the protein an overall positive charge. DNA has a negative charge, and so it gives it a chance to stick. Now, people have noticed for a long time that when you take a non-dividing cell and you stain the nucleus, the inside of the nucleus has different levels of light and dark areas. And so, since we knew it was chromatin, biologists a long time ago said, well, why don't we call the dark areas heterochromatin, and we'll call the light areas euchromatin. And let me show you a picture of what I'm talking about. So here is the nucleus of a cell, and you can see we have some dark areas. So any little dark spot would be classified as heterochromatin, and any other little light spot we would call euchromatin. That means different. In fact, if we break it down, that's protein colored different. Different colored proteins. And U means good, true, or well. So, true chromatin. Now, since that time, we've now understood what's going on. Why are some places dark and when some places light? The reason the dark places are dark is because the DNA is tightly packed. You can see how tightly packed it is here. In places that are light colored, you can see that the DNA is unwound. This is a fantastic way to regulate gene expression. How hard would it be to transcribe this? Oh, it'd be super hard. Look at that. Can you imagine RNA polymerase trying to get in there? So, we find that tightly coiled DNA usually is the place where genes are inactive, where the DNA is inactive. In euchromatin, this is going to be the part of the DNA that often gets expressed. So, if you want to turn off a gene, what could you do? Coil it up, right? Coil it up. If you want to express a gene or turn on a gene, uncoil it. And it turns out there's ways to do that. In fact, you might notice in this one, I have a lot of little stars. Those are called histone modifications. These are little molecules that are found inside the nucleus that go and attach to the histone proteins that go and cause them to unravel. So if we think about, hey, let's turn on a gene, right? You could actually have a gene that makes those histone modifiers. Right? Those modifiers go and they bond to heterochromatin, open it up. Beautiful. You're halfway there to transcription. If you want to go and close it up, you can just do the opposite. You can go and take them off, and then it'll coil up to its default. So here's a way to regulate genes. Uncoil them to turn them on, coil them up to turn them off. Isn't that cool? In fact, we'll find out that this is the way that what's known as the epigenome actually goes and works. Right? Now, the epigenome means that modifications of the genome can be made not from changes in the DNA sequence of itself, but regulating how those genes are expressed. Let me give you an example. Right? Every cell of your body has exactly the same DNA. Right? But some of your cells went to make a liver. Some of your cells are found in your bone. Some of your cells are found in muscle. They all have exactly the same DNA. So how do we get muscle cell and a liver cell and a bone cell all from the same DNA? It's how, during development, those genes were turned on and what genes were turned off. How those cells differentiated is controlled by differences in the epigenome. And we'll talk more about that when we get to genetics. Okay. Now, when we look at somebody's chromosomes, the 23 pairs of chromosomes that we find in the human body, all right, if we look at a typical body cell, so if we went and looked at one of your heart muscle cells, or we went and looked at one of your fat cells, or if we went and looked at one of your kidney cells, we would find that those cells all have 2 sets of chromosomes. 23 pairs of chromosomes. Now, the reason that we call them homologous, when you see that word homologous, right, that means the same. It's almost like having a complete set, a complete cookbook for a recipe for making all the proteins in the body, and a backup. We get one recipe cookbook from mom and one recipe cooked from dad. And think about these as chapters. So we have, 23 different chapters. Right? So here's chapter 1 from mom. Here's chapter 1 from dad. Here's chapter 2 from mom. Here's chapter 2 from dad. Right? So we get one of each pair from mom and one of each pair from dad. 23 homologous pairs. Now, this condition of having 2 sets of chromosomes in a cell is called a diploid condition. Diploid means double. Right? So this is double. So it's almost like having one complete set and then a backup set. Now, there are cells out there that just have a single cookbook. They just have one set. These are things like egg cells and sperm cells. The sex cells just have one set of chromosomes. And for that, we call them haploid. Haploid cells. So, this is body cells. These are sex cells. Now, to blow your mind a little bit, we find out that in other organisms, for example, if you eat a mushroom, all right, like a fungus, right, you're going to find you're not eating diploid tissue. You're eating haploid tissue. In fungi, all right, most of the body is composed of cells that just have one set of chromosomes. And it turns out there's just a tiny few cells within a mushroom that are like this. And they only last a short, brief amount of time. So it's a topsy-turvy world. And if you look at plants, it's even worse. There are plants. Right? Sometimes you have 2 generations of plants. You have sort of the larval stage and then the adult stage. And we often find that the young stage, the larval stage, is haploid. And the adult stage is diploid. Especially with primitive plants, especially when we look at things like mosses and ferns and things like that. A lot of seedless vascular plants have very, very strange haploid and diploid systems. Now we know that right before cells divide, they replicate their DNA. And so every one of these will make a copy of itself. So we'll get a copy of the one from mom and the one from dad. A copy from the one from mom and the one from dad, and so on and so forth. And that's what we're seeing here. So here's the one from dad, here's the one from mom, right? And as you can see, we have a homologous pair. Let's say that's chromosome number 1. So we have chapter 1 of our recipe book from mom and a chapter from dad. And right before cell division, they replicate. Now, a very interesting thing happens. Remember, this is just one long piece of DNA. So it makes a copy of itself, and that copy stays attached. So here, let's say this is the original, and that's the copy. And they stay attached to each other. We call that a replicated chromosome, and this an unreplicated chromosome. We can also call that a duplicated chromosome. That's an unduplicated chromosome. You notice the one from mom does this too. And so the original and the copy are held together by a beautiful little protein called cohesin. I like that because it sounds like cohesion, like binding to like. Now in this little picture you might say, oh, okay, there's a little cohesin here. Turns out that cohesin often runs the length of the arms. And so they're often stuck down the length of the arms. So that's what happens after replication. Right? So in our body cells and in our sex cells, we'll see this happens there too. All right? When a cell is fixing to divide, first thing that'll happen is the DNA will replicate. And those chromosomes, all right, will become replicated chromosomes. All right. Now, next week is fantastic, especially if you're a Thanksgiving dinner fanatic. All right? I never was. All right? To me, turkey dinner is pretty boring. In fact, I'd much rather have lasagna or Mexican food or Indian food than turkey dressing and cranberry sauce. All right? But I know a lot of Americans, this is their favorite holiday because they get to eat their favorite stuff. Now, when I was a kid, my parents, all right, would cook Thanksgiving for us and our extended family, and they would begin the night before Thanksgiving. They would probably go and start cooking that evening, the night before Thanksgiving. And then the next morning, they would get up really early. They sometimes would start at 6. And they would cook till about noon. All right? And then we'd eat about 1 o'clock. So they would put in, 8 to 10 hours of preparation for the feast. And when it came down to eating, we were done in about an hour. Right? So for every hour we ate, there were 10 hours of preparation. Right? So it turns out it's like that. In fact, let me get the math right. The same thing happens with cell division. It takes 90% of the time to prepare for the actual separation of the chromosomes, for the separation of the cytoplasm, for the pinching in two. Right? 90% of the time it takes to prepare for that, and then just 10% of the time to actually go and do the division. Right? So it's just like preparing Thanksgiving and then eating Thanksgiving. So all the preparations and the eating, right, analogy works great for what we call the cell cycle. The cell cycle is all the preparations and the final cell division. So let me show you what the cell cycle looks like. Here we go. Now the actual cell division, where we divide the nucleus and divide the cytoplasm, occurs here in what we call the M phase. M for mitosis. And that represents just 10% of the time. The other percent of the time is all the preparations that it takes to go and do that. Oftentimes, when we go and we eat something or watch something or participate in something, we don't see all the preparations that went into it. One of my favorite documentaries is about a world-class sushi chef. He's in his 80s in Japan. He is incredibly famous in Japan. And it costs to go to his restaurant, which is very small. He only has a couple of customers every night. It costs thousands of dollars. I mean, it's almost like buying a car to go and eat there. Now, if you look at the amount of effort it takes just to produce that meal every day, it's amazing. And he says 95% of what it required to actually go and give you this spectacular meal, you never see. Right? You only get to see the very end of it. And it's just like the cell cycle. Now, the first part of what we call interphase. Interphase are all the preparations. All the preparations, just called interphase. The first phase of interphase is what we call the G1. Now you might be wondering, why did they call it G1? Well, G is short for gap. When we were first looking at interphase, right, we knew that there was a gap in time from the end of the physical cell division of a cell to the time where the DNA replicated. So there was a gap in time. And then there was a second gap in time after the DNA replicated and the physical division of the nucleus and the division of the cytoplasm occurred. And so they called this gap number 2. When I was a student taking biology at Villa Park High School way back in the 1970s, my biology teacher, Mrs. Heddick, told us that during interphase, the cell was resting. We now know it's not really that way. The cell is incredibly active. It's like watching my parents go and prepare Thanksgiving. A lot of stuff going on. So in the first phase, we have lots and lots of metabolic activities. In fact, this is often what we call the primary growth phase. In the second phase here, this is when the organelles replicate. Now imagine what you're doing is you're going from one house and you've got to make 2 identical houses. If you have 2 identical houses, all right, each house has to have furniture, it has to have appliances, it has to have carpeting, it has to have lighting. All the stuff that was in that one house, you have to have enough for both houses. And so in this case, in G2, that's where the replication comes in. So we get a lot of organelles duplicating. So that's where the ER comes from. That's where the Golgi apparatus comes from. That's where the ribosome's coming from. This part right here is probably the most important part of this interphase. And this is what we call the S phase. S stands for synthesis. Remember, synthesis just means to build or to make. So this is synthesis as in DNA synthesis. So the S phase, DNA synthesis. This is where the DNA makes a copy of itself. This is where we go from unduplicated chromosomes to duplicated chromosomes. Now, in your body, there are lots of different tissues that are not constantly cycling. These are not places like your skin and your digestive tract and things like that. For example, if we look at your brain, or your spinal cord, or your nerves, if we look at your skeletal muscle, if we look at your heart muscle, you're going to find that those have left the cell cycle. And what we mean by that is they don't divide anymore. That's why spinal cord injuries and brain injuries are so devastating. That's why muscle atrophy is so devastating. Because once those cells are gone, they're not replaced. In fact, I can only think of one type of muscle that can undergo cell division, and that is found in the uterus. When a woman is pregnant, the smooth muscle that's found in her uterus actually goes and increases in size by 16 times. A normal uterus is about the size of your fist. So imagine having a Hulk fist with a full-term pregnancy. That's the only muscle that I know of that actually goes and divides. And eventually, over time, a lot of those cells die off when it shrinks back to normal. In fact, you can tell the difference between premenopausal women who've had children and ones who haven't. Of course, the uterus is going to be larger in the ones that had children. But for the most part, we find that a lot of things like muscles and nerves, certain types of organs, never get replaced. They actually end up in what we call the G0 phase. And let me show you what we mean by the G0 phase. Once they leave the cycle, they go out here. Right? They never go back and keep going through the cycle. So G0 phase. Now, some of these cells can be triggered to go back in. A great example of this, we used to, sometimes when people would get in car accidents or have a hard fall, sometimes they would rupture their spleen. Your spleen is here on your left side, just next to your stomach. And it's about fifth side, but it's a little bit flatter. And oftentimes what would happen is when they ruptured their spleen, they would have a lot of hemorrhage to go into and internal bleeding. So they often would go and do splenectomies, where they would go and take them out. But we find that in children, in fact, kids under 12, if there's just a little piece of the spleen left, it will regrow and regenerate. So what's happening is those cells have left the cell cycle, but the loss of the neighboring cells has triggered them to go and re-enter the cell cycle and continue dividing until it regenerates a spleen. When we think about cell division, the most important part that we want to really think about is the whole point is that we want to make sure that every daughter cell has DNA in it, has a full endowment of DNA. Now for somatic cells or body cells, all right, that's going to be that every daughter cell is going to have 2 sets of chromosomes. They're going to be diploid cells. So this is a nice little picture. This is from a previous textbook that I had, but I loved it. And what you're seeing here is mitosis, just focusing on what the chromosomes are doing. So here is chromatin, and you might recognize, hey, that's the nucleolus right there. But right before cell division, then what happens is the chromosomes coil and supercoil. Right. Then the chromosomes migrate to other parts of the cell, right, to the both ends. You can see your little metaphase here, and then it separates. We have 2 separate cells now, and eventually the chromosomes go back to their chromatin. But each daughter cell now has the exact complement, exact endowment of what was in the parent cell. Now, how long does this process take? It varies according to the cell, but let me give you a couple of examples. The fastest ones we know, the fastest cell division in animals, comes from fruit flies. Right? We know a lot about fruit flies. You can go from an egg to an adult in 2 weeks. Right? So you would imagine that since their lifespan is so short, they've got some pretty rapid cell division. And we do. For example, if we look in the embryo fruit fly cell embryos will go and divide every 8 minutes. That's amazing. That's amazing. Think about all the replication. So that is one quick cell cycle. A typical mammal cell, that includes us, takes about 24 hours. So one of your skin cells, to make a new skin cell, takes about every 24 hours. To make a new intestinal cell, 24 hours. That's how long the cell cycle is. Liver is amazing. Liver are incredibly slow. Right? To have a liver cell divide, it takes more than a year. This oftentimes happens. Sometimes you'll have people who have cirrhosis of the liver or degeneration of the liver, and you have cells there that are dividing to kind of keep up, but because their division is so slow, the losses quickly outpace the gains. And the most variation that we find in length is that length of the gap 1, of the G1 phase. All right. Now, one of the best ways to look at cell division is to look at something where the cells are really big, that there's a beautiful contrast between the nucleus and the cytoplasm, and that it's easy to tell what's going on. And probably one of the best examples or models for seeing cell division is to look at plants, particularly what we call the root tips of plants. And what you're looking at here is the root tip of an onion. Right? If you've ever had, for example, a green onion and you see the little tiny roots coming off, well, you can snip those off, do a longitudinal section, put them on a microscope slide, stain them, put a little cover slip on, and that's what you get to see. Because at those little root tips, that's where the mitosis is going on as they grow. And as you can see, there's a variety of different stages that you see in the cell cycle. Most of these, for example, are in interphase. You'll notice, in fact, if you look at those little dots, you go, wow, look at all the nucleoli. Remember, nucleoli, even though you look at drawings, usually more than one. Usually more than one nucleoli. Right? And then you'll start to see ones where you go, well, look at that. That one looks a little bumpy there. This is something that has just entered prophase. So this is interphase. And this one where the chromosomes have started to coil and supercoil. Right? And then eventually we get something crazy like this. They coil even more. And then you get things like this. And then they split apart. Isn't that wild? So, what we're going to talk about are these different stages that they go through. Now, part of the cell cycle, if you remember, part of the cell cycle is interphase and then the M phase. So we're going to go and start with our cell cycle here. So we have interphase, and then we have what we call the M phase. Actually, M phase is short for mitotic phase. Now, we already know what happens during the M phase, the interphase. All right. Here we have an interphase. We have our nucleolus. The DNA has copied, so we can say that the S phase has already occurred. Now, since interphase lasts 90% of the time, when you look at a slide of cells in mitosis, 90% of the cells you're going to see are going to be, oh, interphase, 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 interphase. You can see if you look around, most of them are an interphase. As a TA used to tell me in entomology, my first semester of entomology, and I'd bring in some bug that I was convinced was rare and unusual, and he'd just look at me and go, huh, common as dirt. Interphase is common as dirt. It's everywhere. It's the easiest one to find. Easiest one to find is interphase, 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 interphase. All right. Now, when the chromosomes start to coil, they look a little bumpy there. All right. And what you're looking at is the beginning of our next phase. This is called prophase. All right. So this is really the first stage of mitosis. Good old prophase. Now, the big thing that happens in prophase, you can see that our spindle fibers begin to form. They're coming from what we call the centrosome. You can see the nucleus starts to fragment, the nuclear envelope. But the big thing is the chromosomes appear. You can start seeing the chromosomes, something you couldn't see when it was chromatin. Now, these spindle fibers do kind of a cool thing. As they grow out, eventually they will go and connect to the centromeres of the chromosomes. So as they grow out, they'll go and connect to the little skinny waist of the chromosomes, what we call the centromeres. And in mitosis, we'll find that they will attach to both sides. So one will go and attach to one side, and one from the other one will go and attach to the other ones. And these will eventually go and migrate so that they're on opposite sides. So a spindle fiber will attach to this one, and a spindle fiber will attach to the other side. And this is what it looks like. Right? So we have our little spindle fibers. Remember, spindle fibers are just microtubules. But they don't attach to the chromosome directly. They attach to a protein that is attached to the chromosome. And this little protein is called a kinetochore. A kinetochore. Remember, kinetic means movement. In fact, I'm kind of curious about that word. I'm going to see if it's in my handy-dandy Tabor. Lardacious. That was funny. All right. Kinetochore. C-K-I-N. See if it's there. Kineta. There it is. But it doesn't have a word. Huh. Well, kinesis means motion. Maybe we can find what core means. C-H-O-R. Cholera. There we are. Ah, here we are. No, I can't mean that. I have to look that one up again. The only thing I found that was close was to dance, like Huntington's chorea, but I don't think that's the case. The dance mover. I don't think so. All right. Now, what we find is when I was a student, both in college and in high school, after prophase came metaphase, right? Well, turns out there is a stage between them. And it's the stage where the spindle fibers actually go and physically attach to both sides. So when the spindle fibers have finally attached to the chromosomes, that begins what we call prometaphase. So as the spindle fibers are growing during prophase, once they connect to the centromeres, then we have what's called prometaphase. So as you can see in prometaphase, the chromosomes are now have these spindle fibers attached to both sides. Now this is a little bit difficult to see in our little specimens, but it is a nice thing to know. Now, so in prometaphase, as you can see, we have our, and what they're doing is that once they attach, then these little spindle fibers, those microtubules can assemble and disassemble, will actually go and pull and push these chromosomes toward the midline of the cell. We often describe that as the equator of the cell. So when the chromosomes arrive at the equator, then we say that it's in metaphase. I like to say that I never met a phase I didn't like. I sure can. Let me just write this first. Metaphase. I'll head backwards here. So the chromosomes are attached to microtubules at the kinetochores. And the chromosomes move to the equator of the cell. All right. So here we are at metaphase. And that seems like a good place to stop. So we'll finish here at metaphase. And I'll talk about metaphase next time, and anaphase, and telophase, and cytokinesis. And we'll talk a little bit about probably my favorite. I don't know. I love the DNA stuff. I love the mutation stuff. But probably my second favorite is meiosis. I just love meiosis. So we'll talk about that starting on Monday after the holiday, which is coming up. Any questions on what we covered today? Okay. Pro means before. Yeah. So before the phase. And meta, I've wondered about meta because it means beyond. It does. Beyond, they're right? Yep, beyond. Yeah, Bed Bath & Beyond's pretty meta. I like that